Hello, my name is Richard Krause. I'm with USGS Great Lakes Science Center, and it's a great honor to be here today for the Lake Erie Committee meeting in 2022 and present some of the research I've been involved in looking at Cisco and lake whitefish habitat use in Lake Erie. We've got a lot of collaborators. Some of the key ones for the information today are listed across the top. And I just want to give a special thank you to the Lake Erie Committee. Each of your agencies has contributed resources to this work, as well as uh, support for proposals in order to get some of this work started. And I'm just very grateful for that. Thank you. I think a good place to start is to remind ourselves how productive the fisheries were in Lake Erie at the turn of the last century. They really were phenomenal, one of the most productive freshwater fisheries of all time, primarily supported by Cisco and to a lesser degree lake whitefish. So Coregonines were king in Lake Erie during this time. But we know this isn't the case anymore. Sometime around the 40s and the 50s, due to overfishing and lots of environmental problems, what we saw was the loss of Cisco in Lake Erie. They're now extirpated. Whitefish still have a population that sustains a fishery in Lake Erie. You can see the recent landings in the plot on the bottom right, but even with lake whitefish, they're a fraction of what they used to be around the middle of the last century. So let's summarize the current situation for both species. For lake whitefish, we have concerns about a low population size and inconsistent recruitment. As far as the fish community objectives go, what we want is conservative management to provide a sustainable harvest. And the big issue here with whitefish is the bycatch in the walleye fishery, because we need to get a handle on that in order to keep the Marine Stewardship Council certification. For Cisco, of course, they're extirpated, and as far as the fish community objectives go, what we want is to consider rehabilitation only as opportunities arise. There are a lot of concerns. The big ones are about whether habitats are unsuitable in the lake and the limited hatchery resources that are already devoted to other priorities. For both species, we have a number of habitat concerns. These are big and ecosystem level conditions here. Biotic aspects of this are the invasive species, for example, mussels, bithotrephes, rainbow smelt. We also have an increase in the past couple decades of harmful algal blooms, and we need a better understanding of predator-prey interactions and how they may influence the life cycles of both of these species. For the abiotic side of things, of course, we have temperature concerns. Climate is changing this. It's increasing the temperature in the hypolimnion during stratification, and hypoxia, which has increased in recent decades, is also reducing the amount of habitat available, specifically in the Central Basin. There are other factors as well that are changing: lake level patterns, hydrography of the lake, as well as the amount of ice cover, and there's various pollution concerns. One of the things, especially if we're focusing on habitat, that I want to make a plug for is acoustic telemetry as a tool for this. It's great for maximizing information from a small number of fish, and we have that situation with both of these species. It also leverages resources that are already in place with GLATOS, the Great Lakes Acoustic Telemetry Observation System. And it can provide the spatial and temporal information on scales that are relevant to fishery management questions. But the basic things we're looking at here are: where do these fish go, when, where, and how do they die? So the approach here is simple, but it's different for each of these species. For lake whitefish, we've been tagging for several years in conjunction with multiple agencies, and we're going to focus on 2 years, 2017 and 2018, when we had the most number of fish detected on an array that was more widely distributed spatially across the lake. For Cisco, we're talking about tagging. I'm going to present results from tagging that happened this year, and releases that were off of Dunkirk, New York, and Huron, Ohio, 50 each of these fish that were reared up to age 4 at the Tunison Laboratory of Aquatic Sciences. What's interesting is, compared to lake whitefish that only had ID tags, these tags reported temperature and also had a device that would be triggered whenever one of these individuals was eaten and the digestive acids eroded a small little window at one end of the tag. That caused the tag to create a different signal. One final thing I want to mention is that Especially for the lake whitefish, what's really important is that we teamed up with the NOAA folks at the Great Lakes Environmental Research Lab to predict at all of the receivers across the lake the conditions on the bottom of temperature and dissolved oxygen so that we could index that to when and where lake whitefish were detected. On this slide is an overview of the detection data from both species. I'm going to play a small animation for each one of them, and you can see that playing right now for lake whitefish on the left. There are interpolated detections in the blue dots, and then the actual detections in the red dots. And you can see these fish moving across the lake on a seasonal basis across 2017 and 2018. High favorability for the southern shoreline, and especially during the summer, high favorability for areas in the region of the Ohio-Pennsylvania border as well as off of New York. And there are also some interesting patterns of rapid movement across the lake to the spawning grounds where they were tagged on western basin reefs. We can contrast this with Cisco. You can see that there was some movement away from the areas in which they were released off of Huron and off of Dunkirk, New York. We have a much shorter data set here. It's just from this past year, May, June, and July, with a little bit of information from August and September. The big thing is there's a gap here that's going to be filled in 2022 when we get out and tend the receivers. Just had to do with the timing of the release of these fish with respect to when receivers were downloaded. The other thing that's important, especially for Cisco, is that in the Central Basin, on various receivers, we were able to deploy data loggers that recorded right near the bottom oxygen and temperature conditions. And so we can index that to detections of Cisco in a similar manner to the way that we can index detections of lake whitefish with the hypoxia model forecast values. On this slide, what you see for both species are some summary plots of the detection data with the habitat data. For lake whitefish, let's take a look at what we have here when we match the hypoxia forecast model temperature and dissolved oxygen with detections, or just looked at the overall distribution of oxygen and temperature on the bottom across the whole entire array, what we're calling habitat availability, and what's shown in the background is the heat map. So for habitat availability, the areas of highest concentration are shown in the red and the yellow and represent these cold oxygenated waters that you see kind of mainly in this swath right here. This combines the July, August, and September data from both 2017 and 2018. And on top of this, we see a contour plot that shows what we're calling habitat use. This is the subset of conditions that were actually utilized by lake whitefish, or the subset of conditions where lake whitefish were detected across the array. And you can see that there's not a complete correspondence. In fact, the areas of high habitat use are shifted slightly towards more oxygenated habitats, less than 15 degrees C in most cases. And so this shows a disproportionate selection of the available habitats. The interesting thing here too is that, and this is mainly from 2017 data, we see some habitat use of lower oxygen conditions and warmer conditions than where these main centers of concentration are. We can contrast this with Cisco. We don't have the same type of data here, but what we do have are detection data that were matched with the data loggers that were placed on some of those receivers. We had 4 fish that spent a lot of time that gave us a lot of good data. You can see these centers of concentration are mainly here and here by the heat map, where we have basically a trend showing that when dissolved oxygen was lower, bottom temperature was a little bit higher, but in general, most of the bottom temperatures were less than 13 C. We're still filling in these data and will as the season progresses and we get more data back from Cisco. But one of the things that's interesting are these dots, which are the temperatures transmitted by the tag, not the ones recorded by the data loggers, but these are tag temperatures plotted against the dissolved oxygen value that was matched with the detection, the dissolved oxygen by the data logger matched with the detection of the fish. And what you see is that when the dissolved oxygen was lower, the temperatures reported by the tag were skewed higher than what the data logger was observing. So this may suggest that these fish are suspending above the bottom. Here's where the presentation diverges a little bit because we don't have complementary data for both species. So on the left with lake whitefish, we don't have anything that gives us information about whether these fish were eaten. But we do get some information on tagging survival because of the port sampling that happens in Ontario. And this is giving us a unique data set complementary to a study on walleye that we published by Skunyan et al. In 2017. And we looked at the healing of the tag wounds and learned something about the long-term survival of these fish and the retention of the tags. Over here on the right side with Cisco, one of the interesting things is we did get some predation signals from the tags, so we do have some confirmed predation events that occurred. One of the things, again, that factors in here big time is the gap in data that we hope to fill in, because as time progressed, since these things were released, we had fewer and fewer fish at large with which to do this analysis. But what we do know, at least preliminarily here, is that 25% of the fish we released were eaten in the first 2 months. And we expect to be able to refine this in much greater detail as we get more data from 2022. Now we can flip it back the other way and look at something that we have for lake whitefish, but we don't have for Cisco. One of the things that we got really interested in with the lake whitefish data was trying to understand if conditions were poor when the fish was detected, where did they move? And we modeled this with a linear model to try to look at how much oxygen on the bottom changed whenever the oxygen was low or whenever the oxygen was high, and all the points in between. So in other words, we wanted to figure out if the fish were responding to low oxygen levels on the bottom. And we also wanted to know if this was modified by temperature because we had that information as well. So what you see here is a plot splitting out the data by year, and what it does is it developed a model to look at the starting dissolved oxygen concentration when the fish was detected, and then based upon the subsequent detection after that event, how much did that oxygen level on the bottom change? So what's interesting here is the overall pattern for both years is that when the starting dissolved oxygen level was low, less than 5, we saw, except for this particular case here where we have a lot of variability, but in both years we saw that on average, the next move indicated an increase in dissolved oxygen, a positive change in the concentration of dissolved oxygen on the bottom. That happened in 2018, that happened in 2017. It was modified a little bit by temperature, but that starting dissolved oxygen concentration was the best predictor of the change in bottom dissolved oxygen concentration between a pair of detections. Now what's interesting is that this trend continued on across the scale of dissolved oxygen. And so when dissolved oxygen was high, we actually saw an average decline in dissolved oxygen on the bottom between a pair of detections. So we're still looking into what this means. We actually think that over here, when the starting value was low, that there certainly appears to be some kind of avoidance, but there may be a different type of process taking place when dissolved oxygen was high that resulted in these fish moving to an area that had lower dissolved oxygen, but still normoxic on the bottom. And finally, I want to talk about the future and ongoing work on both of these species. For lake whitefish, recently, in the past couple of years, we've been able to deploy data storage tags. And we have to get these back in order to retrieve the data, so I want to say thank you to the port sampling program in Ontario. I've already provided some tags. We just got these tags back. We've downloaded the data. It adds a vertical dimension to habitat to help answer some of the questions that I raised earlier. And you can see just a snapshot of the summary data here showing a lot of variability in temperature and depth. Looks very interesting. It coincides with movements across the lake, movements for spawning, as well as what the fish is doing during stratification. On the Cisco end of things, I want to say thank you to GLaDOS and Great Lakes Fish Commission for coming up with some extra tags to tag the remaining Cisco that still had disease certification at Tunison Laboratory of Aquatic Sciences. Getting those fish tagged now, and releases are planned for this spring. At this point, I want to leave you with three main take-home points to reiterate from the presentation so far. First of all, we have this interesting habitat use pattern geographically along the southern shoreline of Lake Erie. Especially during summertime, we can see the southeastern area of the lake is really important for these fish, and we need to look at this in greater detail. As far as the oxythermal habitat use goes in the summertime, we definitely see that there's habitat available, but also that they utilize areas with low oxygen, or at least where there's low oxygen on the bottom. So we need to investigate further the vertical element of habitat use combined with the horizontal. As far as the directional movements go, we definitely can start to look more closely at the avoidance of hypoxia and high temperatures and how fish choose their habitat, how they move when they encounter these conditions, and this may allow us to simulate different scenarios in the future. What are the implications for fishery management? For Cisco, we're definitely getting new and direct insights on the predation mortality and the habitat use during stratification that will help address some of the outstanding questions that bear on rehabilitation. For lake whitefish, we're learning more and more about when and where they're vulnerable to fishing and how the oxythermal conditions in the lake can modify this. And finally, in a related way, the Great Lakes Water Quality Agreement has nutrient reduction goals that, if they're successful, will change the trophic status of Lake Erie. And what we can do with the data that I've presented here is start to understand how achieving these goals may impact the management of fisheries. And at this point, I'm happy to address any questions you might have.